Okay, so welcome to this next video in which we are discussing adrenoreceptors. Okay, right. So we're in the process of discussing the beta-1 adrenoreceptor on the heart, and we're trying to understand how the activation of the beta-1 adrenoreceptor causes an increase in the force of contraction of cardiomyocytes. Okay, so we're just looking at the process of excitation-contraction coupling, which is the mechanism by which an action potential which is occurring on the cardiomyocyte's membrane triggers contraction of the cardiomyocyte. And the way it's going to do this is by turning the electrical signal into a calcium signal that's within the cytoplasm, which we know will then bind to troponin C, and then that troponin C will cause a conformational change which moves the tropomyosin so that it no longer blocks the active sites on the actin filament, and the myosin filaments can now begin climbing up the actin filaments and causing contraction of the sarcomeres, which will then cause contraction of the entire cardiomyocyte. Right. Okay, so we've discussed that the way that excitation-contraction coupling occurs is that the action potential, when it arrives at this portion of membrane within the T-tubule, will then cause the activation of L-type voltage-gated calcium channels. These will allow a little bit of calcium in from the extracellular fluid into the cytoplasm, and it will go into this little gap between the membrane of the cell and then the membrane of the SR within the cell, which is known as the dyadic cleft. And what's emerging is this concept of a calcium synapse, where the peripheral cell membrane is viewed as like the presynaptic membrane, the dyadic cleft is viewed as analogous to the synaptic cleft, and then the SR membrane is viewed as analogous to the postsynaptic membrane. Okay, so this little bit of calcium that comes in through the L-type voltage-gated calcium channel, that's called a calcium sparklet, and basically, even though we're going to have calcium sparklets occurring all over the membrane of the cardiomyocyte, they're not big enough to add up into a calcium signal that's large enough to actually cause that much contraction of the cardiomyocyte. So what we need is amplification of the calcium signal. And what's going to happen is the calcium that comes in through these L-type voltage-gated calcium channels is going to activate type 2 ryanodine receptors in the SR membrane. These are going to open and release calcium from the lumen of the SR, and this calcium that then comes out of the SR all over the place will then be the main part of that calcium signal that then causes the contraction of the cardiomyocyte. Okay, so let's go back to our discussion of ryanodine receptors. Okay, so we know that ryanodine receptors are tetramers, and basically they're made up of subunits that look like so, okay, with 6 membrane spanning alpha helices and a P-loop in between the 5th membrane spanning alpha helix and the 6th membrane spanning alpha helix. And each of these subunits for ryanodine receptors are around 5,000 amino acids long, which means that this tetramer of ryanodine receptor subunits will have overall around 20,000 amino acids within it. These structures are enormous. They are giants of the protein world. You can see them on electron micrographs. Okay, they are massive, massive protein complexes. Okay, now, basically, we've discussed how ryanodine receptor subunits only form homotetramers. So, for instance, you can combine 4 ryanodine type 2 receptor subunits together to make a type 2 ryanodine receptor, and that's the form of ryanodine receptor that you have present within the heart. Okay, so type 2 ryanodine receptors made out of 4 of these type 2 ryanodine receptor subunits. Okay, right. Now, I just want to discuss other proteins that the type 2 ryanodine receptors are complexed with, okay, that are within the membrane. So, basically, each type 2 ryanodine receptor is complex with with 2 other proteins that are also sitting on the SR membrane. So every single one of these 4 ryanodine receptor type 2 subunits will have 2 other proteins associated with it, and I want to show these proteins now. Okay, so I'll put them here and here. Okay, so they span the membrane a single time, okay, and then on the SR luminal side, they associate with another special protein. So this box here, this is supposed to represent another protein. So let me highlight these in. So we have one protein here, and we'll let this represent junctin. Okay, so this protein that spans the membrane a single time, this is called junctin. And the other one, which also spans the membrane a single time, which I'm now colouring in blue, this can represent triadin. Okay, so basically each type 2 ranitidine receptor subunit within the type 2 ranitidine receptor tetramer will be associated with a junctin protein and a triadin protein. And now both of these proteins, both junctin and triadin, on their SR luminal side, are associated with a protein called calsequestrin. Okay, so this is calsequestrin. And basically calsequestrin binds to calcium ions. So you have calcium ions bound to calsequestrin, multiple calcium ions bound to calsequestrin. And the advantage of this is that when these type 2 ranitidine receptors open, you will basically have lots of calcium ions stored very close to the type 2 ranitidine receptor. And it will be these calcium ions that are bound to the calsequestrin that will go through the pore and be released into the cytoplasm, basically. Okay, so basically what's going to happen if we go back to this picture is the calcium is going to diffuse across the dyadic cleft. It will bind to the type 2 ranitidine receptor subunits, and it causes the opening of the type 2 ranitidine receptor channel. Okay, so this is a phenomenon known as calcium-induced calcium release, where the calcium that comes in through the L-type voltage-gated calcium channels, the calcium that we've called a calcium sparklet, is going to induce more calcium release. So this is calcium-induced calcium release. And for short, calcium-induced calcium release is often abbreviated to C for calcium, I for induced, C for calcium, and then R for release, kicker. Okay, so CICR stands for calcium-induced calcium release. Okay, so what's going to happen is these type 2 ranitidine receptors are going to open and they'll release calcium out into the cytoplasm. Okay, and this calcium signal that you get coming out of the type 2 ranitidine receptors, this is then called a calcium spark. So calcium level is going to go up around the type 2 ranitidine receptor, and that signal is called a calcium spark. Okay. Meanwhile, the calcium concentration in the SR lumen is going to go down, and the reduction in calcium concentration around the type 2 ranitidine receptor in the SR lumen is called a calcium scrap. Okay. So it's basically, you take the calcium spark, you take the park portion of spark, and you spell those letters backwards, and you get calcium scrap. Okay, right. So calcium spark is the rise in calcium concentration in the cytoplasm around the type 2 ranitidine receptor. A calcium scrap is the decrease in calcium concentration around the type 2 ranitidine receptor on the inside of the SR. Okay, right. And now what happens is all of these calcium sparks sum together to then create you a global calcium signal, so that calcium goes up in the cytoplasm of the entire cardiomyocyte, whether it's an atrial cardiomyocyte or a ventricular cardiomyocyte. And I should just say, in the case of atrial cardiomyocytes, all of the calcium synapses will be at the peripheral membrane rather than on the T-tubules in that case. So you'll still have these calcium synapses, and they'll still produce these calcium signals, calcium sparks, calcium sparklets, and calcium scraps. Okay, and that produces a global calcium signal that then activates the troponin C, which then leads to the movement of tropomyosin, so that the myosin filaments can now climb up the actin filaments and contraction can commence. Okay, right. So we now want to see how stimulation of beta-1 adrenoreceptors on the surface of a cardiomyocyte, so here's our beta-1 adrenoreceptor, how is this going to lead to an increase in the force of contraction? Well, basically, the idea is this. Usually when a cardiomyocyte contracts, you get some sort of calcium signal, so you get a certain rise in the level of intracellular calcium in the SR. Okay, now I should just say I want to take a step back for a moment because I've forgotten to tell you about one important thing, okay, which is how do we reverse the calcium signal? Okay, so at the moment what we've got is if we plot calcium concentration against time, okay, so let's have calcium concentration versus time. What we will get is when an action potential arrives, calcium concentration in the cytoplasm will go up like so. But to make the cardiomyocyte relax before we then have another contraction, we need to then return this calcium back into the SR. And to do that, basically, you have proteins in the SR membrane that we've met before, known as the SERCA pump. Okay, and specifically the form of SERCA that you have within cardiomyocytes is SERCA2. I won't give a specific splice variant. SERCA2, but okay. So basically, there are multiple forms of SERCA, and the form that you have within cardiomyocytes is called SERCA2. Okay, now what does SERCA2 do? Well, it pumps 2 calcium ions into the SR lumen, and then it pumps 3 protons out of the SR lumen, so out come 3 protons. And every time it does that, it hydrolyzes ATP to adenosine diphosphate and inorganic phosphate. Okay, so basically it's an active transport system that returns calcium into the SR lumen. So once the action potential has passed over the cardiomyocyte, then the L-type voltage-gated calcium channels will shut. They'll stop releasing calcium, so the calcium sparks will stop. Then also, what I should say is that the type 2 ranitidine receptors show a similar phenomenon to the IP3 receptors. Okay, so at low calcium concentrations, they are activated by calcium. So the calcium that's coming from the calcium sparklet will activate the type 2 ranitidine receptors. It will open and release a large amount of calcium. However, when calcium gets too high in the cytoplasm around the type 2 ranitidine receptor, that will negatively feed back on the type 2 ranitidine receptor, and the calcium then causes it to close. So type 2, ranitidine receptors overall and IP3 receptors are actually very similar. Okay, they're both activated by low calcium concentrations and then inhibited by high calcium concentrations. Okay, right. So basically, when calcium gets too high around the type 2 ranitidine receptor, they will become inactivated and they'll close. Okay, so now we're no longer getting any more calcium coming into the cytoplasm. Then what will happen is this SERCA2 pump here will start returning the calcium into the SR and removing it from the cytoplasm. Okay, and that's an active transport process. So calcium will then go down, basically. And then you'll wait a little while for the next action potential, and then when the next action potential comes, you'll get the whole thing occurring again. Okay, and this is every time the heart beats. Right, so every time the heart beats, you release a certain amount of calcium into the cytoplasm, and what does the calcium do? It will go and activate sarcomeres. Now my question to you is, do you think every sarcomere will be activated to the full extent that it could be activated? I.e., do you think at normal physiological levels, okay, so at the normal amount of calcium that we are releasing before we have stimulated the heart to beat faster, do you think that every single active site on the actin filaments are being exposed? Okay, do you think that every troponin molecule is moving, is getting a calcium ion bound to it, and is moving tropomyosin out of the way, and therefore the myosin filament has the optimal amount of climbing that it can do? Well, the answer is no. Okay, so not all sarcomeres are going to be working to their full potential, because the calcium signal that you produce at normal sort of resting levels when the heart's just beating at rest, okay, the calcium signal that you produce isn't large enough to actually activate every troponin molecule so that the tropomyosin is completely moved out of the way of all of the active sites. So some of the active sites will remain occluded, so you won't build up as much contractile force as you could. So basically, if you want to increase the force with which the heart contracts, what you can do is increase the calcium signal, because if you increase the calcium signal, then more troponin molecules are going to be activated. The tropomyosin is going to be moved out of the way of more active sites, so you're going to have more active sites of the actin filament exposed, and then the myosin filaments will be able to climb up the actin filaments more optimally, and therefore you're going to get more cross-bridge cycling occurring and more force being produced. Okay, so more contractile force. So you can increase the force of contraction of a cardiomyocyte by increasing the calcium signal, and the reason is that this is going to recruit a greater number of cross-bridge cycling events occurring, okay, because more active sites of the actin filaments will be exposed. Right, so we want to now see how beta-1 adrenoreceptors are going to lead to an increase in these calcium spikes that are going to occur in the cardiomyocyte. So what they're going to do is basically turn them into something like this, okay, so that you get a larger calcium spike with every heartbeat. Okay, and this is occurring in every cardiomyocyte. They'll all be producing greater contractile forces, so the whole heart's going to beat with a greater force. Okay, right. So how does it do this? Well, basically, we have seen that the beta-1 adrenoreceptor will cause the activation of the GS heterotrimeric G protein, which will then cause the activation of adenylyl cyclase enzymes producing cyclic AMP, which will activate protein kinase A enzymes. The catalytic subunits of protein kinase A are now going to phosphorylate and activate 3 targets that are going to be important. So, firstly, let me just draw this picture out again. So we'll have our T-tubule here. Okay, so the first target is going to be the L-type voltage-gated calcium channel. Okay, you're going to phosphorylate the L-type voltage-gated calcium channel. After all, that's what protein kinase A catalytic subunits do. Okay, so you're going to get phosphorylation of this L-type voltage-gated calcium channel here, and what that's going to lead to is it's going to increase the conductance of the L-type voltage-gated calcium channel. Okay, so we're going to target the L-type voltage-gated calcium channel here. So this is the L-type voltage-gated calcium channel, so that the overall result of that is that the amount of calcium that the L-type voltage-gated calcium channel lets in is going to be increased. So the size of the calcium spark that you produce is going to be greater. Now, what does this mean? Well, it means it's probably going to be capable of activating a greater number of type 2 ranitidine receptors. Okay? Not only does this sort of contribute to the global calcium signal in itself, but the main thing that this is going to do is it's going to recruit more type 2 ranitidine receptors. Okay? Then, the next target is going to be the type 2 ranitidine receptors themselves. Okay, so here is type 2 ranitidine receptor. I'll draw another one here. Okay, so in the membrane of the SR, we have the type 2 ranitidine receptors here, and basically this increased calcium means that we're going to recruit more type 2 ranitidine receptors, but in addition, protein kinase A is going to phosphorylate the type 2 ranitidine receptors as well, so this is what protein kinase A is going to do. And again, it's going to increase the conductance of these type 2 ranitidine receptors here, so that the amount of calcium they're going to release into the cytoplasm is going to increase. Protein kinase A phosphorylation can also modify the inactivation properties of the type 2 ranitidine receptors. So remember I told you that basically at low levels of calcium, type 2 ranitidine receptors get activated to open, but when calcium gets too high around them, that causes them to close. Basically, if you phosphorylate the type 2 ranitidine receptors, they will stand a larger concentration of calcium around them, basically. So it will take a larger concentration of calcium around them in the cytoplasm to actually cause them to inactivate. So it desensitizes them to the inactivation by calcium. They will still inactivate due to calcium, but it will require a greater concentration of calcium around them to actually do that. Okay, so Basically, what this overall means is that you're going to get more type 2 ranitidine receptors activated, and you're also going to get a greater amount of calcium actually released from each of the type 2 ranitidine receptors. So overall, the size of the calcium spark now is going to go up. So that's how we end up increasing the concentration of calcium that we produce in the cytoplasm every time the heart beats. Now, finally, protein kinase A also phosphorylates the circa 2 pump. Okay, over here is circa 2. So basically, when you decide that you're going to release a greater amount of calcium into the cytoplasm, you also have to take account of the fact that now we need to pump all that calcium back in every time in between the beats of the heart, okay, so that the heart can relax back down. Okay, so the cardiomyocyte also needs to be able to relax. So we're going to have to activate circa 2 so that circa 2 is going to be capable of pumping this increased amount of calcium back into the cytoplasm in the same amount of time, basically. Okay, so protein kinase A will also phosphorylate and activate the circa 2 pump so that it's ready to deal with the increased amount of calcium that we release into the cytoplasm on every beat of the heart, basically. And this will overall then result in these increased spikes. So it will get higher because we've activated the L-type voltage-gated calcium channels and the type 2 ranitidine receptors, and it will drop down back to 0 in the same amount of time because we have activated these circa 2 pumps as well. Okay, and that is going to lead then to more sarcomeres being recruited, more cross-bridge cycling being recruited, because you're going to have now more active sites on the actin filaments exposed, and that will then lead to a more optimal cross-bridge cycling, basically, so that you have at any one time more myosin cross bridges interacting with actin, and therefore more force being generated. Okay, right, so I just now want to finish our discussion of beta-1 adrenoreceptors with some pharmacology then, so with some drugs. Okay, so basically, let's start off with some agonists for the beta-1 adrenoreceptor. Okay, so let's say this is our beta-1 adrenoreceptor here. Here's the amino terminus extracellularly, and here's the carboxylic acid terminus intracellularly. Okay, so we know that agonists for the beta-1 receptor include adrenaline and noradrenaline. Okay, however, we want to see exogenous agonists. So a selective agonist for the beta-1 adrenoreceptor is a drug known as dobutamine. Okay, so dobutamine specifically binds to the beta-1 adrenoreceptor and increases the force with which the beta-1 adrenoreceptor will contract. Increases and activates the beta-1 adrenoreceptor, and the beta-1 adrenoreceptor is mainly expressed on the heart, so dobutamine results in an increase in heart rate and also in an increase in the force of contraction of the heart. So dobutamine is actually used in heart failure, because in heart failure what happens is the heart just becomes too weak, basically. The contraction force that the heart is capable of generating is too weak to actually pump blood around the body, okay, and that's known as heart failure because the tissues of the body don't get enough oxygen and nutrients to service their needs, and therefore the heart is effectively failing the body, so it's called heart failure. So dobutamine is used in heart failure to increase the force with which the heart contracts to try and make sure that the heart pumps enough blood around the body to actually service the needs of the peripheral tissues. Another example of an agonist for beta-1 adrenoreceptors is isoprenaline. Now, isoprenaline is not a selective beta-1 agonist as dobutamine was. Isoprenaline is actually an agonist for both beta-1 receptors and also beta-2 receptors, okay, whereas dobutamine is selective for the beta-1 adrenoreceptors. But again, isoprenaline will result in activation of the beta-1 adrenoreceptors on the heart, which, as we've seen, will result in an increase in the frequency with which the heart beats, so an increase in heart rate, and also in an increase of the force with which the heart contracts. Okay, now some antagonists for the beta-1 adrenoreceptors. Okay, so antagonists for it, which are drugs, remember, which will bind to the receptor at the same site as noradrenaline or adrenaline would bind, but do not activate the receptor. So they bind there and block other ligands from being able to bind, but they themselves do not activate the receptor. So a selective antagonist for beta-1 adrenoreceptors is atenolol. Okay, a non-selective antagonist for beta-1 receptors is propranolol. Okay, and propranolol is also an antagonist for beta-1 and beta-2 receptors, so it's a non-selective beta antagonist, and these sort of drugs would often be referred to as beta blockers. And they can be used in tachycardia, basically. So if you've got a dysrhythmia, okay, which just means some sort of problem with the beating of the heart, so some problem with the pattern of the beating of the heart. So usually in a healthy heart, the heart beats every second or so, and it propagates down the heart in the beautiful way that I have shown you when we discussed the cardiac cycle. However, if the way that the heart beats isn't that beautiful, if there's something wrong with it, maybe if it's beating too fast, or maybe if the actual conduction pathways are all weird, basically. Maybe you have the ventricles contracting before the atrial or something like that, or at the same time as the atrial, something terrible like that. It's called a dysrhythmia. Okay, now an example of a dysrhythmia is when the heart is beating too fast, and that's called tachycardia. Okay, and basically if you've got tachycardia, and I'm not talking about physiological tachycardia, so if you go jogging, your heart will beat faster. That's physiological, it's supposed to. It's because the oxygen and nutrient demands of your peripheral tissues have gone up, so the heart needs to pump more blood around the body in a certain amount of time. So that's a physiological tachycardia. Pathological tachycardia means tachycardia if you're sitting down on a seat, basically doing nothing. Your heart's beating far too fast. That's a pathological tachycardia. Okay, and basically these drugs, atenolol and propranolol, beta blockers, can be used to treat that, basically, because they will bind to the beta-1 adrenoreceptors and they'll stop noradrenaline from being able to stimulate the beta-1 adrenoreceptors. Hence, they'll reduce beta-1 stimulation on the heart, and we've seen that beta-1 stimulation is responsible for the increased rate at which the sinoatrial node fires action potentials. Okay, so if you block that, then you're going to decrease the rate at which the sinoatrial node fires action potentials, and hence you're going to decrease heart rate. Okay, so there is just a little bit of pharmacology. In the next video, what we'll move on to is looking at the beta-2 adrenoreceptors.